Hello, all. My name is Austin D. Clement. I will be discussing the paper called A Role for GLUT3 in Glioblastoma Cell Invasion that is not recapitulated by GLUT1. This paper was published in the journal called Cell Adhesion and Migration by Catherine J. Libby and others. So, glioblastoma. Glioblastoma, or also known as GBM, is a lethal malignant brain tumor that is complicated to treat. Targeting the glioblastoma's metabolism process might be effective in treating the tumor's potentiality for cell invasion. The use of glucose transporters, GLUT, can play a major role in cancer metabolism. So in the paper, there's GLUT1 and GLUT3. Glucose transporter 1, also known as GLUT1, is responsible for glucose movement across the blood-brain barrier, also known as BBB. While glucose transporter 3, GLUT3, is responsible for the uptake of glucose into the neuron. GLUT3 has shown to have a five-fold higher affinity for glucose than in GLUT1. There has been no research to suggest that GLUT3 has a role in GBM processes. However, GLUT3 has been shown to play roles in other cancerous areas of the body, such as breast, colon, liver, lung, head, and neck. So the hypothesis of the paper. The hypothesis of the paper is that GLUT3 has a role in mediating glioma invasion outside of its role in metabolism that is mediated by the C-terminal end of the protein. So there's several methods and materials that was used in the paper. And as one may notice, that on the right side are the methods and materials, and these were the titles. Each one is the title of the method and materials section of the paper. Also, each of these titles goes into depth. So when you all read the paper, each of the titles, and you go in each of the titles, you will see that there's a specific procedure for each one. We will not be necessarily discussing that due to time constraints and the nature of the methods and materials section. But there's some things to highlight, and we're not going to be highlighting, for instance, in the cells in GBM patient-derived xenografts, things such as the university in which the D456 or JX22 was obtained, things of that nature we don't have to discuss, nor the nitty gritty of the methods and materials section. Just some things to highlight that you might want to be aware of. For part of protein visualization for GLUT1 and GLUT3, the use was the Proto Online interface to find the visualizations of both of those, GLUT1 and GLUT3. Also, as well as for the protein alignment here, the use of NCBI, BLASTP, and the FASTA sequences was used. The gene overexpression, that was self-explanatory, I believe, along with the mRNA extraction, cDNA generation, and qRT-PCR. So things like that, explanatory, again, like the Western blotting and the antibodies. The measurement of cell growth I guess the best or like the most important thing to take out of that was the crystal violet staining and solubilization of it. That would be probably the, I guess, the best part of it, or the most one that you had to highlight. Things of that nature. The flows, this part, the self sorting that was used, but self-explanatory. Boyden, chamber invasion, and migration. That was very much used for the invasion and migration part of it. But again, for the most part, self-explanatory. Glycolytic, glycolytic stress tests. I found it was interesting, that part for the seahorse. It talked about that, but again, that isn't that's self-explanatory, along with the RNA sequencing analysis and data set analysis. All of that could easily be self-explanatory and looked up fairly easily. So that's the methods and materials section. So as you may notice that this each slide for the results section is broken up with the title, and the title is each title of the results section is the title that corresponds with the title designated in the paper, so that everything is maintained its integrity when discussing the paper and discussing like the charts or the graphs and the experimentation. And as you can see on the right, this is the graphs that this is the layout of the graphs and the experimentation. And on the left is the results or the synopsis that was obtained, or just an overall explanation of what was obtained through the results and through the graphs and through the visualizations. So now we can go and get started and dive into, well, figure one. And this is figure one, and this is called GLUT3 is elevated in invasive GBM cells, and that's what was the focus on. That was the focus on figure one. So it, in figure one may notice So in figure one may notice that in figure 1A, that, well, actually in figure 1A to figure 1B, this is the SA. So as one may notice, in figure 1A, the x-axis represents the bulk tumor and the leading edge. And this is as it pertains to the SLC2A3, which is also GLUT3. And then on the y-axis, one may notice a relative expression. And then in figure B, and this is as it compares to figure 1A and figure 1B, that in figure 1B, again shows y-axis relative expression, x-axis relative represents bulk tumor and leading edge, but in figure 1B, this relates to SLC2A1, which is also GLUT1. So one may notice that 1A and B, that the leading edge of GBM noticeably increase in GLUT3 expression rather than in GLUT1. Or one may say, and showed an elevated the leading edge of GLUT3, the leading edge of GBM was noticeably elevated in GLUT3 of GLUT1. In GLUT1A, in GLUT1C and in graph 1C and graph 1D, one may notice the deep white matter invasion. This is as it relates to the deep white invasion. In 1C, it represents GLUT3 expression for the y-axis. And on the x-axis is a no and yes. In graph figure 1D, as it relates to deep white matter invasion, the GLUT1 mRNA expression is on the y-axis, no is on the x-axis, along with yes being on the x-axis. The conclusion was that greater correlation of deep white matter invasion in GLUT3 than in GLUT1. On to figure 1E. This shows the control GLUT1OE, GLUT3OE, and this is as it relates to V5 and actin. And the conclusion was this was a confirmation of GLUT1 and GLUT3 expression. As it relates to 1F through 1H, we'll be looking at for 1F, the control of the D456, the N1G, and this is all relates to D456. In 1G, we see the GLUT3. In 1H, we see GLUT1. And this is as it relates to D456 and the overexpression of GLUT3 and GLUT1 and the control. And we find, and then now for F through H, we see that the overexpressed GLUT3 over here, the GLUT3 cells showed an increase invasion than both the control and GLUT1. So as it relates to and this is as it relates to D456, but in graph 1I, and this as it relates to D456 invasion, we see that y-axis represents percent invasion normalized to control, and on the x-axis we see the control, GLUT1, and GLUT3. 1I, the synopsis was that the overexpressed GLUT3 D456 cells had a 50% increase than the control. In figure 1J, as it relates to D456 migration, we see that the y-axis represents percent migration normalized to control. And on the x-axis, we see the control, GLUT1, and GLUT3. In figure 1J, the synopsis was that there's no significant difference in migration. In figure 1K, as it relates to D456 growth, we see the y-axis represent relative growth normalized to control. And on the x-axis, we see the control, GLUT1, and GLUT3. The synopsis of 1K indicate an overexpressed GLUT1 D456 cells had a slight increased growth. Again, the title of this is match the same title as the title in the research paper in the results section that matches this graph or figure 2. The title is Elevated GLUT3 Expression Promotes GBM Invasion in Vitro. So as it relates to figure 2, we see that figure 2A through 2C, this is all, these two figures represent, 3 figures represents the invasion. And also, this is as it pertains to JX22 or JX22 GLUT overexpression. So, as it relates to that, we see that in figure 2A, this is the control as it relates to invasion and the relationship between JX22 GLUT overexpression. In figure 2B, we see GLUT3 as it relates to the invasion and JX22 GLUT overexpression. And then in figure 2C, we see GLUT1 as it pertains to JX22 GLUT overexpression and invasion. The synopsis of these 2A through C was that the overexpressed GLUT3 JX22 cells showed an increased invasion than the control or GLUT1. On to 2D. Figure 2D as it relates to JX22 invasion, the y-axis represents percent invasion normalized to control, and the x-axis represents the control, GLUT1, and GLUT3. The synopsis of this graph showed that there's an overexpressed GLUT3 JX22 cells showed roughly a 200% increase. As it relates to 2E, and this is as it relates to JX22 growth, the y-axis represents relative growth normalized to control, and the x-axis represents control, GLUT1, and GLUT3. 2E results show that there's no significant difference in growth between the two, the two GLUT1 and GLUT3 proposed. So on to 2F. And the synopsis of 2F and 2J have been combined. And this is as all relates to JX22 GLUT3 sorted. So in 2F, this is as it relates to JX22 unstained. The y-axis represents the count, And the x-axis represents the APC-CY7-A. Again, in figure 2, 2G, as it relates to JX22 anti-GLUT3, the x-axis represents APC-CY7-A, and the y-axis represents the count. In figure 2H, again as it pertains to JX22 GLUT3 sorted, GLUT3 is low. This is GLUT3 low. And in figure 2I, this is GLUT2I. This is GLUT3 high in 2I. So in figure J, we see as it relates to JX22 sorted invasion that the y-axis represents invading cell count, and the x-axis represents JX22 GLUT3 low and JX22 GLUT3 high. The results of 2F through J indicates GLUT3 high cells were more invasive than GLUT3 low cells. So now we are on to figure 3. Again, the title, the intracellular C-terminal tell of GLUT3 is necessary to induce GLUT3-mediated invasion, relates back to the same title given to the figure in the paper. So as one may notice, when comparing group 3, figure 3A and 3B, in 3A represents GLUT3, the protein, and in figure 3B represents GLUT1 in the protein, and one may notice that the regions, the two main regions, different, as indicated, both in a box-like structure within the areas of difference. And this area of difference is shown very much so in figure 3C with the slightly different color oranges or different color orange within the chain, and the different color purple within the C-terminal end of the chain as well. So figure 3A through C shows that the GLUT3 and GLUT1 proteins are 80% similar, but there are two regions of difference, and one being the C-terminal tail. In figure 3D, one may notice that there's GLUT1 and GLUT3, both in its what is considered normalized spot, and this is just a representation of the chain and representing what's being cut out and what's being put back in, or what's being switched. And that's what's going on in 3D. In 3D, we see this switch and we see GLUT1, GLUT3, and now we see the chimeras of each one. So the chimera that we see is GLUT3-GLUT1 CT, which means that the GLUT3 structure is being the structure from GLUT3's C-terminal is being placed with that of GLUT1's C-terminal, and the same with GLUT3-GLUT1 EC. We see instead of the C-terminal being removed or switched out to the GLUT1, we see the EC or extra cellular region being switched out as well. So this is just that the differential regions of GLUT1 will combine with that of GLUT3. In figure 3E, we see that the GLUT3 is there in the plot, and GLUT3-GLUT1CT is there, and GLUT3-GLUT1EC is there. And this is as it relates to V5 and tubulin. And this is just a confirmation of GLUT3 and GLUT3-GLUT1CT and GLUT3-GLUT1EC expression, just to make sure that they're all being, to confirm that they're all being expressed. Now on to the figure 3F to 3I. In 3F, we see GLUT3 as it relates to D456. In figure 3G, we see GLUT3-GLUT1CT as it relates to D456. And in figure H, in 3H, we see GLUT3-GLUT1EC as it relates to D456. And now we see in figure I, or 3I, as it relates to D456 invasion, the y-axis represents invasion, percent invasion normalized to control, and the x-axis represents GLUT3, GLUT3-GLUT1CT, and GLUT3-GLUT1EC. And we can take from through figure 3F to I that there's no significance in GLUT3-GLUT1EC D456 cell invasion. However, GLUT3-GLUT1CT showed 60% decrease in cell invasion. So for figure J, we see as it relates to D456 growth, the y-axis represents the relative growth normalized to control, and on the x-axis we see GLUT3, GLUT3-GLUT1CT, and GLUT3-GLUT1EC. In figure 3J, there was showed no significant difference in D456 growth from the chimeric proteins. As it relates to 3K and 3L, we see in 3K that this is GLUT3 as it relates to JX22. And then in 3L, as it relates to GLUT3-GLUT1CT, this relates to the JX22. In figure 3M, we see as it relates to JX22 invasion that the y-axis represents percent infestation normalized to control, and the x-axis represents GLUT3 and GLUT3-GLUT1CT. In figure 3N, we see as it relates to JX22 growth, the y-axis represents relative growth normalized to control, and the x-axis represents GLUT3 and GLUT3-GLUT1CT. Figure 3K to N, the synopsis was that JX22 invasion from the chimeric protein GLUT3-GLUT1CT has shown a reduction while growth had no significant change. So in the next one, this is figure 4. Again, the title of this corresponds with the title of the figure in the paper. So the title is called "The Intracellular C-Terminal Tail of GLUT3 is Necessary to Induce GLUT3 mediated invasion. So the experiment that was conducted again, but in reverse manner, and a focus on only GLUT1 GLUT3 CT. So everything stayed the same. Well, the procedures stayed the same, but instead of focusing on GLUT3 and having GLUT3 have the features of GLUT1, it's the reverse. So it's now GLUT1 having the C terminal end of GLUT3 to try to see if there's going to be the increase in invasion. And it was actually shown, yes, that was correct. As it relates to, and again, in 4B, and that was indicated in 4A, the switch of the C terminal end of GLUT3 onto GLUT1. And the 4B just shows a confirmation of expression of GLUT1 and GLUT1 GLUT3 CT. In figure 4C, we see GLUT1 as it relates to D456, and in figure 4D, we see GLUT1 GLUT3 CT as it relates to D456. And we see that again, GLUT1, and I'm purposefully skipping the E and F. We'll come back to it. GLUT1 in 2G in 4G, we see here as it relates to JX22, and in GLUT1 GLUT3 CT as it relates to JX22 in figure 4H. We see that here. Now we see that C and D are similar. CD are similar to GH in the increased nature by visualization. Now, as we go back to 4E, we see as it relates to D456 invasion, the y-axis indicates percent invasion normalized to GLUT1, GLUT1, and on the x-axis we see GLUT1 and GLUT1 GLUT GLUT1 GLUT1 as it relates to GLUT1 GLUT3 CT, and then in 4F as it relates to D456 growth, the y-axis represents relative growth normalized to control, and on the x-axis it represents GLUT1 and GLUT1 GLUT3 CT. Now we see that deep in 4E that GLUT1 GLUT3 CT had an increase in cell invasion. However, in D456 growth, there was no real significance. Same thing was shown as it relates to JX22 invasion and JX22 growth. The y-axis in figure I shows that the percent invasion normalized to G or GLUT1, and the x-axis represents GLUT1 and GLUT1 GLUT3 CT, and that showed an increase in cell invasion similar to that of E, or in its nature. And then in J, one may notice is JX22 growth. The y-axis represents relative growth normalized to control, and the x-axis represents GLUT1 and GLUT1 GLUT3 CT, and one may notice that there's little to no growth. So extracellular matrix, so the next title of this is figure 5, called Extracellular Matrix Organization Gene Signature is Altered in GLUT3, Overexpressing D456 GBM cells. So the in 5A, one may notice that from the results that there's on the y-axis is the Go terms, that and the Go terms meaning that gene ontology terms that were found, and this is as it relates to pathway enrichment for GLUT3 versus GLUT1. And on the x-axis, one may notice the fold enrichment, which is between 2.5 and a little bit over 12.5. The colors indicate the negative log 10 and the p-value, while the size of the dots represents the gene number. So the top three terms that were found were that the low density lipoprotein, particle receptor binding. The next term was the extracellular matrix organization. And the third term was the retrograde protein transport, ER to cytosol. So on to 5B, and I know the time, so we're going to just briefly go over this. The Go terms, as stated before, are over here. Then the next category is the fold enrichment. The next category is highest p-value. The next category is up regulated. And the next category is down regulated. Now, in 5B, one may notice that the extracellular matrix organization has the highest number of transcripts out of the three. And so, and that's because of the, I'm sorry, as it will, so, and there's in 5B, certain transcripts include CD44, COL1A2, COL3A1, ITGA3, LAMB3, SPARC, SPP1, and BCAN. So on to 5C. This is as it relates to TCGA-GBM correlation, and this is for 5C, D, and E. So on the on the y-axis is the SLCA3 relative mRNA expression, and on the that's the y-axis, and on the x-axis there's the SPP1 relative mRNA expression. This was shown to be positively correlated. We're going to skip all charts due to time constraints. Next is 5D, y-axis being SLC2A3 relative mRNA expression. The x-axis being CD44 relative mRNA expression. This was shown to be having a positive correlation. In 5E, the y-axis is SLC2A3 relative mRNA expression. On the x-axis, there's a ITGA3 relative mRNA expression. This was also shown to have a positive correlation. And as it relates to 5F, again, we're skipping the charts. As it relates to 5F, the y-axis represents SPP1 mRNA expression. And on the x-axis represents the control, GLUT1, and GLUT3. And this is as it relates to D456. And it was shown for the synopsis was that SPP1 mRNA expression was increased in D456 cells overexpressing GLUT3. Summary of the results. D456 GLUT3 overexpressed cells increased invasion. D456 GLUT1 overexpressed cells decrease invasion while increase growth. JX22 GLUT3 overexpressed cells significantly increased invasion. GLUT3-GLUT1-CT showed 60% decrease in cell invasion, and JX22 GLUT1-GLUT3-CT overexpressed cells increased invasion. SLC2A3 GLUT3 had a positive correlation with SPP1, CD44, and ITGA3. And this is just the summary of the results that I found to be the most prominent. Conclusion. The authors stated that the results indicate a direct and distinct functional role for GLUT3 in mediating cancer cell invasion that has of yet not been reported to our knowledge. I would say that that's a very bold statement to make, and I wouldn't say it seems a little too definite. So the results indicated a direct and distinct functional role of GLUT3. I would say there needs to be more research. Before making a statement that big, I would just suggest more research need to be made, and then one can make that statement. I would have to look at other articles or other research articles in the future to see if that statement could be made. Just not by this project, I believe that statement could be made. Next is the reason for GLUT3 overexpressing cells invasive nature originates from its C-terminus. They did find that. More drugs should be developed and tested to target GLUT3 and its C-terminal tail. And lastly, this research could be used to help treat other cancers that GLUT3 is involved in, such as breast, colon, liver, lung, head, and neck. So the dream, how would you utilize the discoveries of this paper? So I am very, very much new to biotech. So from my knowledge that I got from, I guess, college, which was last week, I would have to say that for the investigation on the role of GLUT3 need to be made, and to better understand GLUT3 and all of its properties, because it seems like there's still more that could be understood around GLUT3 and what it could potentially do in the health field. Also, there needs to be a focus on ways to cleave and replace the C-terminal tail of GLUT3 within the brain. And lastly, finding ways for people to be experimented on in a clinical trial setting, I think would be necessary, because to my knowledge, GBM is a rare disease. And since it's a rare disease, I think one would need to really take a lot of time to understand the disease. And from my knowledge, because it's a rare disease, it's hard to find the best way to treat it. And it's hard to find different modes of helping those who have it and are afflicted with it. So I think research needs to be really, if this is going to be a potential research project, really understand, and it is a research project, but really understand what is the disease and just understand it from a very holistic outlook, and find ways in which one could be able to treat people having those diseases in real life and actually being applicable. And I think that's going to be key to helping others who are afflicted. Next question is, could you build a new project or even a whole new company out of these findings? I would say no because it's very specific. Also, it's a rare disease, and because it's rare, I don't think it's necessarily profitable in that way. And yeah, you can't really make a nonprofit out of it or anything like that, or even an actual profitable company because it's so rare. But if it was on other cancers, I could see it most likely happening in that way. Or like if you try to think of it like, oh, you want to be a small company and then become a big company like Genentech, I could see that happening. But I don't think you could necessarily make a whole new company out of these findings alone because it's super specific and the number of people afflicted with the disease are rare and small in population. So, could you build a new project? Yes, I believe you can. You could again like create projects and treatments to investigate other cancers involving GLUT3. That's a very much a easier and faster route. But if you're trying to do a long-term project and potentially make a new company out of it in the long run, I guess you could create a predictive model that assembles possible variables that could be attributed to glioblastomas, because then you can use that same predictive model in other diseases that are rare. So you could find those how to treat those rare diseases as well. So that's one way of looking at it, and that's one way of possibly getting a whole new company out of it as well. I would like to just thank you for your time.